This lesson, we will review TEKS B.8B. Predict possible outcomes of various genetic combinations using monohybrid and dihybrid crosses, including non-Mendelian traits of incomplete dominance, codominance, sex-linked traits, and multiple alleles. This video is part 2 of a two-part series for TEKS B.8B. Part 1 reviews Mendelian inheritance and monohybrid crosses, and Part 2 reviews dihybrid crosses and non-Mendelian inheritance. In Part 1 of this lesson, we learned how to make predictions about monohybrid crosses and Mendelian traits. Also, we learned how to identify an organism's genotype and phenotype for a single trait. For example, a pea plant that is homozygous dominant for pod color has the genotype G, G, and a phenotype of green pods. A pea plant that is heterozygous for flower color has the genotype P, lowercase p, and a phenotype of purple flowers. And a pea plant that is homozygous recessive for seed shape has the genotype lowercase r, and a phenotype of wrinkled seeds. But what would be the genotype of a pea plant that has purple flowers, green pods, and is heterozygous for both traits? Simply put the genotypes of the two traits together. The genotype of this pea plant would be P, lowercase p, G, lowercase g. What if we crossed two of these plants? Would their offspring have purple flowers or white flowers, green pods or yellow pods? To answer this question, we can use a Punnett square. In this Punnett square, we will be modeling a cross between two individuals that are heterozygous for two different traits. This is known as a dihybrid cross. To make a Punnett square for a dihybrid cross, start with a 4x4 grid. We know that each parent will pass its offspring one allele for flower color and one allele for pod color. But which allele? The dominant one or the recessive one? It could be either, so we have to list all four possibilities for each parent. Each parent could pass a dominant allele for each trait, a capital P and a capital G. Or each parent could pass a capital P and a lowercase g, or a lowercase p and a capital G, or lastly, a lowercase p and a lowercase g. That sets up our Punnett square. Now, copy each allele combination into the cells of its row or column. The Punnett square is complete. These are the genotype possibilities for an offspring produced by this dihybrid cross. How do we know which phenotypes result from these genotypes? Remember the relationship between the two alleles. The allele for purple flowers is dominant to the allele for white flowers, and the allele for green pods is dominant to the allele for yellow pods. Using this information, we can identify all of the phenotypes that result from the genotypes in this dihybrid cross. As you can see in the Punnett square, this dihybrid cross results in four possible phenotypes: purple flowers with green pods, purple flowers with yellow pods, white flowers with green pods, and white flowers with yellow pods. What are the probabilities of each of these phenotypes? Remember that in a Punnett square, each box represents an allele combination, and each allele combination is equally likely to occur. In this Punnett square, there are 16 boxes or 16 allele combinations. Let's start with the phenotype purple flowers with green pods. How many allele combinations produce this phenotype? 9. So, in this dihybrid cross, there is a 9 out of 16 chance, or about a 56% chance, that an offspring will have purple flowers with green pods. How many allele combinations produce purple flowers with yellow pods? 3. So, there is a 3 out of 16 chance, or about a 19% chance, that an offspring will have purple flowers with yellow pods. Continuing our analysis, we can see that there are 3 allele combinations that produce white flowers with green pods. So, there is a 3 out of 16 chance that an offspring will have white flowers with green pods. Lastly, there is only one allele combination that produces white flowers with yellow pods. So, there is a 1 out of 16 chance, or about a 6% chance, that an offspring will have white flowers with yellow pods. These phenotype ratios, 9 out of 16, 3 out of 16, 3 out of 16, and 1 out of 16, are the same ratios that Mendel discovered when he performed his dihybrid crosses more than a century ago. So far in this lesson, we have focused on the traits of Mendel's pea plants. But did you know that Mendelian traits can also be found in humans and other organisms? In humans, for example, the allele for having freckles is dominant to the allele for having no freckles. The allele for normal melanin production is dominant to the allele for albinism. And the allele for normal mucus production is dominant to the allele for the disorder known as cystic fibrosis. These are just 3 of many examples of human traits that follow the Mendelian dominant recessive pattern of inheritance. But what about a trait such as eye color or blood type? Do all traits follow the Mendelian pattern? No. There are many other patterns of inheritance found in nature. These are often referred to as non-Mendelian inheritance patterns. One example of a non-Mendelian inheritance pattern is called incomplete dominance. Remember that in Mendelian traits, one allele is dominant over the other allele. Incomplete dominance is different. In incomplete dominance, neither allele is completely dominant over the other. The heterozygous genotype results in an intermediate or blended phenotype. Flower color in snapdragon plants follows the incomplete dominance pattern of inheritance. A cross between a red snapdragon and a white snapdragon does not produce red or white snapdragons. It produces pink snapdragons. The pink color is a blend of red and white. It is an intermediate phenotype. What do you think would happen if 2 pink snapdragons were crossed with each other? We would expect about 25% of the offspring to be red snapdragons, about 50% of the offspring to be pink snapdragons, and about 25% of the offspring to be white snapdragons. Another example of non-Mendelian inheritance is called codominance. Remember that in Mendelian inheritance, when an organism is heterozygous for a trait, the organism expresses only one allele, the dominant allele. How is codominance different? In codominance, when an organism is heterozygous for a trait, both alleles are expressed. A roan cow's spots are an example of a trait that follows the codominance pattern. In the image shown, notice that the roan cow has distinct reddish spots and distinct white spots. These cows are heterozygous for the trait of hair color. They have an allele for red hair and an allele for white hair. Both alleles are expressed, resulting in red and white spots. Next, we will examine an inheritance pattern known as multiple alleles. Remember that in Mendelian inheritance, each trait has only two alleles. Green pods or yellow pods, for example. But not all traits work this way. For some traits, there are more than two alleles. In humans, the trait of blood type has three alleles, A, B, and O. A and B are codominant, and O is recessive. Look at the blood type chart shown. Because there are three alleles, there are 6 possible genotypes. By contrast, a Mendelian trait would have only 2 alleles and therefore only 3 possible genotypes. A fourth example of non-Mendelian inheritance is polygenic inheritance. Remember that in a Mendelian trait, a single gene controls the trait, but some traits are controlled by multiple genes. They are polygenic traits. In humans, eye color, skin color, and height are examples of polygenic traits. Let's examine height more closely. Earlier, we studied stem height in pea plants, a Mendelian trait. There are only 2 possible phenotypes for stem height in pea plants: tall or short. By contrast, when we observe the trait of height in human populations, we see that there is a spectrum or a range of phenotypes. Humans can be short, tall, or anywhere in between. This is typical for polygenic traits. Extreme heights are less probable and therefore less common in the population, as shown in the bell curve graph. And lastly, a fifth example of non-Mendelian inheritance is known as sex-linked traits. To understand sex-linked traits, let's take a moment to remember that DNA is organized into structures called chromosomes. For example, human DNA is organized into 46 chromosomes, 23 from the mother and 23 from the father. One of these pairs of chromosomes is known as the sex chromosomes. They're called sex chromosomes because they determine a human's biological sex. Females have 2 X chromosomes, and males have an X chromosome and a Y chromosome. Just like normal chromosomes, sex chromosomes have genes. For example, in this model, you are looking at a pair of sex chromosomes with a few genes highlighted in different colors. Can you tell if these chromosomes represent a biological male or a biological female? Because they both are X chromosomes, they represent a biological female. Now let's focus on the yellow gene for a moment. Let's say it's a gene that affects vision. The allele for normal vision, represented by a capital B, is dominant to the allele for colorblindness, represented by a lowercase b. So, would this individual be colorblind? No, because this person has an allele for normal vision, and normal vision is the dominant trait. This person would have normal vision. Now, would this work the same way for a male? Remember that the sex chromosomes for a male are not XX, they are XY. In the model shown, notice that a Y chromosome is much shorter than an X chromosome. In fact, Y chromosomes have only a fraction of the genes that X chromosomes have. What does that mean? In this case, it means the Y chromosome doesn't have an allele to offer for this trait. Instead of two alleles, this person has only one allele, the color blindness allele, for the vision trait. Therefore, this person would be colorblind. All it took was one recessive allele to express the recessive trait in a male. In this way, sex-linked inheritance is different from Mendelian inheritance. In fact, we write the genotype for sex-linked traits in a special way. We include the sex chromosomes and the alleles of interest. In this case, the genotype is capital X, capital Y, and the capital X has a superscript of a lowercase b to represent the color blindness allele. What is the genotype for our earlier female example shown here? The answer is shown under the model. Now, what would happen if those two genotypes were crossed with each other? Just as we've done all lesson long, we can make predictions about a genetic cross using a Punnett square. Here is the Punnett square prepared with our two parental genotypes. Now, fill out the grid by copying the alleles into their respective rows and columns. Based on our completed Punnett square, we can make the following predictions about the offspring of these two parents. There's a 25% chance that their child would be female and colorblind. There's a 25% chance that their child would be female with normal vision. There's a 25% chance that their child would be male and colorblind. And there's a 25% chance that their child would be male with normal vision. Now you know how to identify sex-linked traits and make predictions about their genetic crosses. Sex-linked traits, polygenic inheritance, multiple alleles, codominance, and incomplete dominance remind us that the science of biological inheritance is complex. No single pattern, including the Mendelian pattern, can explain all of the mechanisms of inheritance found in nature. But thanks in large part to the work of Mendel, scientists today have reliable models for solving increasingly complex problems in the field of genetics. Let's review the vocabulary of this lesson. A trait is a characteristic of an organism. For example, eye color or pea pod color. An allele is an inherited set of genetic instructions for producing a particular form of a trait, such as green pods. A genotype is an organism's specific allele combination. Homozygous is the description of an organism that has two identical alleles for a particular trait. Heterozygous is the description of an organism that has two different alleles for a particular trait. A phenotype is an organism's expressed traits that result from its genotype. In a heterozygous genotype, if only one allele is expressed in the phenotype, the allele is considered dominant. An allele that is not expressed in a heterozygous genotype is considered recessive. A Punnett square is a tool that displays all of the genotype possibilities that can result from a cross between two individuals. A monohybrid cross is a cross between two individuals that are heterozygous for a single trait of interest. A dihybrid cross is a cross between two individuals that are heterozygous for two traits of interest. Mendelian inheritance is the pattern of inheritance discovered by Mendel through his studies of pea plants. In Mendelian inheritance, a trait is determined by its two alleles, specifically whether the alleles are dominant or recessive. Non-Mendelian inheritance refers to inheritance patterns that do not follow the Mendelian inheritance pattern. Incomplete dominance, for example. Incomplete dominance is an inheritance pattern in which neither allele is completely dominant over the other, resulting in an intermediate phenotype. Codominance is an inheritance pattern in which the heterozygous genotype expresses both alleles. Multiple alleles is an inheritance pattern in which a trait has more than two alleles. Polygenic inheritance is an inheritance pattern in which a trait is controlled by more than one gene. Sex-linked traits are traits that are controlled by genes located on the sex chromosomes. In this lesson, we reviewed TEKS B.8B. Predict possible outcomes of various genetic combinations using monohybrid and dihybrid crosses, including non-Mendelian traits of incomplete dominance, codominance, sex-linked traits, and multiple alleles. This video is part 2 of a two-part series for TEKS B.8B. Part 1 reviews Mendelian inheritance and monohybrid crosses.